hello everybody. Nice to be here and be with you. I can see in the list of people across the top, lots of people I recognise and lots of people we work with quite regularly, so really good to see you all along. So I just wanted to give you a bit of an overview of the work that AHDB does and the people that we talk to and the industry perspectives that we get from that, and then think a little bit about the IPM strategies of managing bacterial pathogens that we could begin to think about. So just a few slides really to think about the problem and what that looks like. So we know that there's over 100 known bacterial pathogens that can affect UK crops, and I just want to give you a picture of a few of them from hort and potatoes. But bacterial problems can also affect cereals and oilseed crops as well. And they're often causing things like soft rots and wilts, and that latter really by sort of blocking xylem vessels and that movement of water. So just a few slides with some pictures of affected crops, and these are thanks to Steve Roberts, who I can see is on the call. So thank you for some of these lovely images that have been really useful for this. But also just thinking about some of the impacts of those pathogens on crop loss that we see in the UK annually. So some really big figures and big impacts from those. So sort of moving on, some more sort of in mushrooms, where we see sort of 0.4 million pounds worth of loss of crops in there. And then as Pippa was just pointing out there, when we look at some of the ornamental crops there, they're kind of, as you say, often not sort of really something that's thought about for the research world, but in the project that we're doing with Steve, there is quite a bit of ornamentals work in there, because we do recognise that these are really important crops, and often those bacterial problems that make them quite unsightly and make them unsaleable cause big problems, and we, as Pippa quite rightly said, we lose them from our garden centre shelves and our gardens, which is really sad. One of the other areas where we see some really quite important and huge amounts of loss are in those prunus species that again Pippa mentioned, but not just the laurel species, which is the picture on the right there, but also in fruit prunus, so the cherry family, where we see those sort of Pseudomonas syringae pathovars that affect that, the Mosphrinorum and the syringae. So in the cherry crops, we're seeing about £6.5 million worth of loss. But in the ornamental crops, which obviously are quite high value crops as well, we're seeing £27 million worth of crop loss in those annually, which is really quite important. And then obviously there are some new pathogens that are coming into there, so things like the Xanthomonas arboricola, which we're sort of is on the horizon. And obviously there's lots of other crops, horticultural crops that are affected, and some of those are by some quarantine pathogens as well, which are really sort of important. Just a quick one on the potato side, obviously things like blackleg affect Bacterium matrosepticum, primarily that, which obviously affects, often seen sort of more than 50% of all seed crops produced in the UK. Estimated losses around that, £50 million annually, just in the UK. So huge impacts on that. So just moving on to thinking about an IPM approach to that. So within AHDB, we're using this prevent, detect, control framework to help us build a robust strategy that encourages farmers and growers to consider all the options available to them for managing crop issues. And we just think this little framework just helps with that sort of a logical approach to management, really. We're also looking at our IPM programme across our different crop sectors around using a sort of production system approach. And that can help us in terms of thinking about if we group those together effectively, we can manage or identify some opportunities for management of those pathogens within that crop, whether they be bacterial or anything else, in similar ways under those different types of production systems. And as Pippa alluded, that sort of protected cropping areas, and those, especially in propagation, can be areas which provide excellent opportunities for spread and development of bacterial pathogens. So I just wanted to sort of start to think about going through some of that framework really, and thinking about the options that are available and that we should be sort of keeping in mind, I suppose. So thinking about prevent and starting off with seed, that's sort of starting off with really clean seed is number one priority really if you're growing anything from seed. So checking how clean that seed is, and if you're, you can speak to your seed supplier around that, but also if you're concerned, there are laboratories that will check sort of pathogen levels on them. Also think about the quality of them, where those seed come from, if they come from a good source that is where the stock plants that are producing that seed are clean and healthy and kept well. But there's also options there to think about sort of treating them if needs be, and we'll come on to that in a little bit later. So thinking about variety selection again, as well, you can think about sort of, even if we don't know that we've got resistant pathogens and resistant cultivars within those some of these species, we do know that there's, within some of them, there's reduced susceptibility to bacterial pathogens. And we know that a lot of growers sort of understand that. Pippa mentioned that some of them aren't growing particular species, but they'll be growing different ones within that crop group, because they know that they're less likely to get those disease symptoms in them. And then thinking about things like cutting material that some growers use. Again, it's thinking about that clean mother stock, thinking about that high health or making sure that that's clean and of good quality, and sometimes thinking about sort of high health production of that through tissue culture, etc. And then on the young plants, thinking about again, where are those young plants coming from? What's the origin of those plants and the quality of them as they come in? What's your experiences of that? I know I've spoken to growers who have stopped buying from some suppliers because they know that they often come in with problems. And they just, they're bringing that problem on site. So what's the reputation of the grower that you're buying from? And then also thinking about sort of quarantining those plants when they come on site, because you don't know what they might be bringing in, and quarantining is just good practice, really. But the prevailing point around prevent is hygiene, and we all know how important that is in terms of keeping stock areas and standing areas of plants clean as much as possible, as well as all the equipment that have been used, pots and all the rest of it. So just moving on to think about detect. So this one's a bit less easy with something like a bacterial pathogen, because obviously it's not an easy one that you could trap spores of or those sorts of things that, as you would with a fungal pathogen perhaps. But what growers can do often, and even sort of amenity growers and home gardeners, is check your crops regularly, walk them and looking for small points of infection, which can spread really quickly in the right conditions. And if you see those plants that are affected like that, just as growers do, farmers and growers do, you need to rogue them out wherever you possibly can. And if there's the possibility of thinking other options to minimise sprays, such as altering the environment if possible, so things like reducing the relative humidity or stopping overhead watering in those crops, if that's the practice that's being used, then that's a strategy that you can introduce if you're starting to see those problems coming in. Pippa talked a little bit about the value of diagnosis. I think for me, there is sometimes value in doing that. I agree entirely. We don't always need to know exactly which pathogen we've got in that crop, but sometimes it's useful to identify that and to identify potential sources of that pathogen coming in. And then the important thing is that if you do detect those problems in your crop, it is to destroy that material, and really that involves things like incineration, for instance, because what we don't want to be doing is just sort of putting that material into composting areas or just leaving it lying around where it can still manage to spread that pathogen around. So finally, thinking about control. So there's obviously been a previous high reliance on copper-based products, which are now, of course, all gone, except for in grapes. There are still a few broad spectrum conventional products around, but areas like sort of elicitors and biopesticides are options, but these are fewer of them, which I'll demonstrate in the next slide. But there's even for those that are available, if you're an organic producer, even access to these is limited, and so that can be problematic. So things like basic substances, so things like calcium chloride, which has been trialled in mushrooms, or acetic acid can also be useful. And then volatiles and terpenes are likely to also have some activity and becoming a bit more present in that sort of biological approach type activity there. And we're trialling some of those within Sector Plus, the main vehicle that AHDB use for our efficacy and crop safety trials. But again, these are quite slow in coming through. So this slide is being kindly loaned to me by Roma Gwynn from BioRationale, and she collated some data on the availability of biopesticidal bacterial pathogens. So you can see that the number of biocontrol products that are produced with fungal pathogen control in mind, compared to those for bacterial diseases, there's a massive difference there and a huge imbalance. And so clearly we need to think about some different approaches for management when those problems do arise, or even to protect our crops. I just wanted to sort of spend a few minutes talking about bacteriophage, which I know that Sarah mentioned in her introduction, and is becoming sort of a really interesting topic, really, I think. So what happens, bacteriophage, for any of you that don't know, I know most of you on the call probably will, but bacteriophage are viruses of bacteria, and we all know huge amounts about viruses now, don't we? So you get different bacteriophage for different bacteria. And they can be isolated from soil, water, plants and animals. They're really the most abundant organism on the planet. So there's 10 to the 31, estimated to be 10 to the 31, which apparently is 1 nonillion bacteriophage on the planet at any one time. So the way that their action and the way that they act is that the phage attach to their target bacterial cell, and using the little sort of what looks like a bit of a coiled spring arrangement in that sort of picture at the bottom of the slide there, they inject nucleic acid into their host, and those nucleic acids multiply up and produce new bacteriophage, which then often form a lysis of the cell, and so it bursts open, killing that cell, and then all of the new bacteriophage are released to repeat the process in other bacterial cells. And one of the great things about them is that they're non-toxic and not pathogenic to eukaryotes, so all the higher plants, and also to many sort of obviously to us and to many animal species as well. They can only be produced in the host, so they're not going to run riot all over the world. Particularly, they're only found in the host because when they are not in the host, when they're not in the host, they can only survive for a few hours. The issue at the moment is that this is relatively new technology for plant protection products. So there's little guidance on the requirements or the ability to navigate the regulatory process. So there's a couple of examples here of AHDB funded work where we've looked at bacteriophage, so one on mushrooms and one on tree fruit there on the cherry. So in the cherry work, a mix of 4 phage isolates were applied prior to leaf 4, and that decreased the bacterial population by approximately 10-fold in comparison to a water control, which is sort of considered comparable to chemical control. And then just finally, thinking about the use of phage in practice. So phage cocktails need to be used, and the reason for that is that limits that resistance development and broadens the host range of those products. You get a co-evolution of the pathogen and the phage. So as the pathogen mutates or changes slightly, so does the phage to keep pace with it. And then they can be used for both prevention and for curative activity as well. Different modes of application there. And then the image on the right shows a post-harvest application of a commercial APS product to reduce soft rots in stored potatoes. So we have to ask ourselves, you know, with the loss of other activity around biopesticides and conventional chemistry, is this a way of managing pathogens in the future? Although the range of projects that are involved in this BPD programme may offer us some more opportunities. Thank you.